I'm teaching the one gene, one protein hypothesis to a group of students who have to study it as part of their syllabus. It's a very difficult concept to teach and extremely tricky for students to grasp, simply because it's so abstract. This lesson tries to grapple with some of these issues in a way that hopefully makes it easy to understand.
Sit. Just sit down. Sit. See, that's what I told you. Long time ago.
Fantastic. How did you know to sit down then? What did you do?
I just had to look and then look.
Did you have any idea what I was doing when you came in?
Were you asking for our attention?
Why did you think I was asking for your attention?
I just looked at the thing and then I saw what you were doing.
So what's the word we could give to what I was doing? What was it? I was signaling in a code. Excellent, because this lesson is all about codes and the secrets that codes can hold within them. Okay. Now the key to any code is knowing how to read it, decipher it. Once you can read a code, you can look underneath it and see what it's actually all about, or the secret message which is hidden within. Codes have been used for thousands of years. The ancient Egyptians used codes. The Nazis in the Second World War used a very famous code called Enigma. Today, we're going to look at a very special type of code. But before we do, we're going to have to use an analogy. It's like a comparison, okay? It's a comparison, so then you can take that thing and compare it to the real one. The reason I'm using an analogy is because the concept we're trying to look at is so abstract and so difficult, it'd be hard just to go straight there, okay? So let's introduce another code. Let's imagine I had a code which was kept within a vault, okay? So imagine over here I had my vault, just there. And the code is projected within the vault. Now, you can't take the code from the vault. Look. I can't get it. I can't get it, okay? But if I can remove that code from the vault and take it to my workshop, I might be able to decipher it. Could I have 3 volunteers, please? Up you come, quickly, and we'll go over to the vault. Okay, have a look what we have there. Can you describe to the rest of the class, if you go into the vault for me, please, and describe to the rest of the class, what do we have in? What's our tools? We have colored paper, we have some pegs,
Rope, yeah.
and we have some rope. Now, they need to get that code to the workshop. Any suggestions how they could possibly do it? Yes, Sean.
They should like get pieces of paper. The first put red.
All right, that's a fantastic idea. Do you understand, Sean, what he said there? So if we had 2 of you holding the line out, okay, 2 of you hold the line out. Okay, should we peg a red on them, please? Okay, Eugenia, what would be the second color we put on?
Would it be red again?
Okay, let's put on red again. So, what I'm going to ask you 3 to do now is, on your own, complete that color code for me, please. So, what we have actually done is made a what of the code? We've made a copy of the code. Very, very good. Now, once we have that copy, our job is to decipher it. We can do that in our workshop, okay? So remove your copy and go and stand behind the workshop for me, please, guys. Now, what we have over here is a supplier. Could I borrow 2 people, please, to quickly come up here for me? That's great. Here we have the supplier. Now, the supplier can supply the codes to the workshop. Could you hold up one of the blocks for me, please? Show them the color code and describe to the workshop what you have there. What's on the back?
1 blue and 2 reds.
3 colors on a code which corresponds to a part. Tony, just come up here for a second, please. Now, your supplier here has parts, okay?
Yeah.
You need to ask for the correct parts. Have a look at the code. It's a 3-color code that you have there.
Okay.
What do you think, Tony? What is the first part that you're going to ask for, please? They've got a three-colour code.
Red, red, blue.
Red, red, blue. Okay. Laura, can you get a red, red, blue from the supplier, please, and take it to Tony for me, please? Very, very quickly. Tony, can you nominate a builder for you? Anybody?
Any of you?
Okay, check the code.
Do you want to come?
Red, red, blue.
Come then, Emmanuel.
Manuel, quickly. Okay. Tony, check the code. Is that correct?
Red, red, blue. Yes.
Okay, give it to your builder. Manuel, can you start building in section 9 for me, please? Section 9. Okay, Tony, next sequence, what's it going to be? Thank you, Laura.
Blue, yellow, red.
Blue, yellow, red. Okay, thank you, Laura. Give it to Tony. Tony, can you check it for me, please?
Blue, yellow, red. Yep.
Okay,
correct.
give it to your builder. That's your second part. Tony, quickly now, what's your third?
Green, yellow, green.
Green, yellow, green. Laura, can you take that over, please?
Thank you. Green, yellow, green. Yep.
Check it. Okay, and give it to your builder. Okay, step back. Okay, the rest of the class. What do we have there, do you think?
A car.
The bit you just did? Is it a car, Remy?
A wheel.
The wheel.
A wheel.
And the back bumper.
Are you sure about that? Yeah. Do we have a full car there?
No.
What do we have? A part of the car. Okay. If we had a separate code, a second piece of code, maybe it'd fill the picture in for us, okay? So let's imagine we're going to do the second part of this sequence, okay? So we're going to do the whole thing again. As you can see, the code has changed. So, without interruption, guys, there you are. As fast as you can, off you go. Where do we go to? Where are we going? Workshop. Fantastic. Round we go. Interpreter. Do your stuff.
Can I have a blue, yellow, red?
Laura, take it over. Emmanuel, area 5, please. And it's going to fit again, okay?
Red.
This one's going to be tricky. Tony, did you check that one?
Yes.
Okay, marvellous. What's next?
Green, blue, yellow.
Green, blue, yellow. Okay, Laura, take it over for me, please.
Green, blue, yellow.
Marvellous. And final piece. Tony.
Green, blue, yellow. Red, green, red.
Fantastic. Fantastic. Give her a round of applause. That's what you call sharpish. Very good. Do you want to take a seat? Okay, marvellous. Do you want to all sit down for me, please? Very, very quickly. Sit down. Well done, Manuel. Good work. Take a seat. Okay. Have a look at the diagram. What do you think we have? A car. Again, do we have a full car? No. No, we have parts of the car. Component parts of the car. Put that in your head, please. Component parts. Okay, now this color code coded for something quite insignificant, really. It was a car. But there are other codes which code for things which are far more important, far more complex. There is a code found within all of us, within our body, within every single living organism, a very special code. Does anybody know the name of it? Yes, Tricia?
DNA.
DNA, fantastic. Can you tell me anything about DNA? What do you know about DNA?
It's a long strand of molecule.
Okay, it's a long-stranded molecule. Where is it found within our body? Does anybody know? Yes,
In our
Ina?
genes.
It's in our genes. And where are our genes found within our body?
In like the cells.
Which cells? Which cells are they found in? Yes, Emmanuel.
All of it except the sexual cells. Which
Is none in our sexual cells?
Now it is in our sexual cells, but I got half of it.
They got half of them. Fantastic. Can I get it out? Yes, Marty.
You can separate. There's a way you can separate the DNA.
Okay.
For example, with a banana, you could get the DNA out of a banana.
Really?
But yeah, but I don't know. Like you could do it out of it.
Well, if I had a lab and £1 million, then possibly I could actually separate the DNA out. I don't have a lab and I don't have £1 million. What I do have is a blender. Now, I've also been to the butcher's and I have some lovely, lovely samples of meat. So I'm going to have a lovely cocktail of thymus gland, which is found just there, and it's not human, no, I promise. I didn't kill anybody in the making of this program. And also some liver, okay? So the first thing we're going to do is I'm going to put it into the blender. Why do you think... Why do you think I have to put it into a blender? Where's the DNA found, did we say? Yes, Melissa?
You have to break it down so you can separate the proteins.
Yeah, very, very good point. I have to break it down so I can separate it, okay? Now, if I just turn the blender on now, what's going to happen?
It's going to fly.
It's going to fly off. It's also going to be rather sticky, okay? So I'm going to dilute it with some water. So I'm just going to add a splash of water to my liver and thymus to make a sort of liver smoothie, okay? Very, very rich in iron. Very, very good for you. Okay, into that concoction, I'm also going to add a few other things. I've got some detergent, and I'm going to add just a little smidgen. Why do you think I need to add detergent? What's it going to do? Yes, Martin?
Separate whatever's left when you put it through a sieve. I have a feeling that that's the DNA.
You're on the right lines, aren't you? You're on the right lines. Yes, Rebecca?
Is it going to separate the fat from like the rest
You're a
of
genius, you know that. It is.
Thank you.
The cell membranes are made up of fat, okay? They've made up of fat. What's the detergent going to do to the fat? What does detergent do to fat? Breaks it down. Very good. So we add a bit of detergent to break it down. Also, I find a little bit of salt helps in this as well, okay? So there we have a little bit of salt, about 2 teaspoons.
A little bit.
There you go. All right. So it's looking good. Now then, what I'm going to do next is give that time to blend. So I'm going to put the lid on and I'm going to switch it on. Ready? Steady. There we go. That's looking nice. That's looking nice. All right, so now we've blended the DNA mixture, we need to add one more ingredient. Now, DNA isn't found sort of just lying about inside the cells. It's raveled very, very, very tightly around proteins. Very tightly. So, to get at the DNA, what we have to do is break down the protein. Now, can anybody remember what is the name of the enzyme which breaks down protein?
Protease?
Protease, fantastic. It is called protease, and it just so happens I have some bacterial protease here. I have to be careful with this because it's a bit of an irritant, but I'm going to add a little bit of bacterial protease to this, okay? What it's going to do is going to break down, there we are, the proteins which is holding the DNA together. Round and round and round we go. That's fantastic. That'll just take a couple of minutes to work. Now, can you see any DNA? No. That's because DNA is actually soluble in water, okay? It's soluble. What does soluble mean? Yes, Tony?
It can dissolve.
It can be dissolved, yes. So what we have is the DNA which is dissolved in there, okay? Now, if I can show you one I've done earlier, just for clarity, here it is. Again, we still can't see the DNA, but the DNA is present in there. Now remember, remind me again, DNA is found as what? A double helix, okay? So DNA is a double helix strand. They're all found within there. Now, what I have here is some alcohol, very, very, very strong alcohol. Although DNA is soluble in water, it is insoluble in alcohol, insoluble. So what do you think I can do to get the DNA out of there? Yes, Sean.
You pour it into the alcohol.
Pour it into the alcohol. Very, very good. Or I could pour the alcohol
Into
the DNA.
Yeah.
Let's have a go at this. Sometimes this works, sometimes it doesn't. What we'll see are two separate layers. Yeah. In the second layer, hopefully, the DNA is going to separate out, and you'll see it either as some fluffy white pillows, or hopefully some strands of DNA. Can you see any strands coming yet?
Yeah.
Can you? Yeah, look at that. That's fantastic. Eugenia, can you get me a cocktail stick out of there for me, please, very quickly? Look at that. Can you see it?
Yeah.
That is actually real DNA, which has been extracted from these thymus glands. So what I'm going to try to do is pull it out. Here it comes. Can you see it all pulling out? It's a bit like snot. But it's far, far more important than that, because that in there contains all your genetic information. DNA. Okay, let's put that away. Where's the DNA held?
In the nucleus.
Fantastic. It's held within the nucleus. So can I have 3 volunteers, please? 1, 2, 3. Up you come. To go into the nucleus. And this time, the DNA code is not a color code. If you can stand in there, the DNA code is a letter code. If you can hold the clothesline for me, please. One over here, one in the middle. Patricia, do you want to come around to peg them on? Okay, using the previous model, we had a red. And what did we put on the first peg? Red. This time we have a C. So what would common sense say to put on there? Common sense would say to put on a C. However, it doesn't work like that. DNA is a double helix. It has complementary bases. Complementary meaning
Things that go together.
things that go together but are not exactly the same. Fantastic. So we're going to have to use complementary bases. So we're not going to use a C. What are we going to put up there? Does anybody know from the structure of DNA? Can anybody remember what the complementary base for C is? Yes, Eugenia?
G.
Fantastic. It is a G. Should we write that down as a rule, please? C goes with G. So it stands to reason that G is going to go with Eugenia.
Does it go to C?
Fantastic. G is going to go with C. C is going to go with G. They're complementary in the double helix. Do we understand that? So what will be the first one up?
G.
Okay, do you want to put it on the first peg for me, please? On the first peg. We have a lot of pegs. Look what he's doing. Is he right?
You're putting a G, so C. CC? Yeah.
Okay, so can you modify that for me then, please? Fantastic. Is he right now?
Yeah.
Okay, keep going. Go on, Tricia. What's the next one? C goes to... Fantastic. T is going to go to A. Wonderful. Well done. So T goes to A. Can you just put the 2 A's on for me and stop there? Now, Laura, a second ago I said to you, if C goes to G, G would go for C, yes?
Yeah.
So if T goes to A, what does A go to?
T.
A goes to T. That makes perfect common sense. And that would be nice and easy and straightforward. Science is never easy or straightforward. I'm afraid we don't have any T's. We're going to have to substitute with a different letter. A always goes to U. Sorry. So, Tricia, A goes to U. Shall we write that rule down and underline it? Because that's a very important one. So, let's have Tricia out here. So, A goes to U. What's the next one? A goes to U. Martin, do you want to take up the slack on the line for me, please? Fantastic. So, we're making a copy of the DNA. Now, remember we said the DNA is a double helix? For simplicity, what we've shown on the board is only one of those strands. Only one of the DNA strands is shown, okay? Because only one of the DNA strands is actually copied. Only one strand is copied. Now, this strand has a very special name. This copy of the DNA has a very special name. Does anybody know what it is? If you like, this strand is a messenger, isn't it? It's a messenger which copies it and takes it to the workshop. But of course, we haven't got a workshop in the cell. What we have is an area called a ribosome. Write that down for me, please. We have an area called a ribosome. On your model, where you've put workshop / ribosome, because the ribosome is a workshop, if you like, of the cell. Okay. Now this strand, which is coming along nicely, as I said, is a messenger. It is sending a message to the ribosome. Okay? So we call this strand messenger RNA, or mRNA for short. You might want to write that down. Messenger RNA, mRNA. Okay, we nearly done? I think that might be plenty. Okay, very, very carefully, let's take it over to our ribosome. So we leave, where are we leaving?
Nucleus.
The nucleus, fantastic. We're leaving the nucleus and we're going over to the ribosome. Here we are. And stand up nice and straight behind and hold it up for me, please. Make sure you don't lose any, 'cause it's very important. Hold it up straight. There you go. Fabulous. So there is our mRNA strand at the ribosome. Is everybody with me so far? Yeah. Shall we walk through again? So we go to the nucleus and we copy what?
The DNA.
Do we copy both strands? Just one strand of the DNA. And what is the copy called?
Complementary.
It's called?
Complementary code.
Complementary bases, excellent, form the what strand? What do we call it? mRNA. And then the mRNA leaves the nucleus. And where does it go to? The ribosome, which works as the what of the cell? The workshop where we construct things, okay? And the last time we used car parts. The ribosome does not use car parts. It uses something called amino acids, which are the building blocks of what? Proteins, fantastic. Proteins. So we have our amino acids. But this time, obviously in our body, we don't have a supplier. What we have is molecules called transfer RNAs. And what does transfer imply? What does transfer? If you transfer something, what does it imply? You switch, you move something, okay? You take something from there over to here. Now, what's missing from our model? What did we have last time? Here.
Someone to carry a supplier.
We had a builder. We also had a person who would decoder. So can I have a decoder here, please? And can I have a builder? A decoder? Yep. Do you want to come across? Now. Watch. It looks really bad, but it's not. Okay? Describe what you see to me. What do you see? Remy, what do you see there?
I see like
In each box.
I see in each box I see a code or something. Which one?
3 capitals.
Yeah, 3 capital
3
letters.
letters. A 3-letter code. Do you remember we had a 3-color code which corresponded to a part? Yes? We have a 3-letter code. What do you think that corresponds to? The mRNA code. So, let's say for instance, what's the first one, Zena?
GGA.
A. Find me GGA, please.
Okay,
Fantastic. Now the part that you need is written next to it. So what would be the part you need?
GLY.
GLY. Now GLY is short for glycine, okay? Which is the amino acid. Now where did I say these are carried by? Who carries these? Transfer RNAs. Can I have 12 of you, please? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Could you come up and be transfer RNAs for me, please? Hold them high and proud above your head like so. Grab one each. Okay, hold them high and proud and just mill about in this area for me. Mill about in this area. Fantastic, fantastic. Just mill about. Okay, freeze for a second. Can I have a bit of space over here, please, so I can give some ribosomes some space? Now then, transfer RNAs are like the supplier. They supply the building blocks. The building blocks being the amino acids. Xena over here, okay, interprets the codes and builds them into the correct order. So, leaving this line clear here, can you go over there and just mill about until you require it? All right, Xena, who do you need first? And call them in for me, please.
GGA, GLY. Anita.
Glycine, where are you? Okay, in you come. Nita, just stand there, please, and hold it for me. Just give it to her. Thank you. Call the next one then, please. AUU.
AUU, which is over here. ILE.
ILE, coming in. Your time is up. In you come quickly. All right. Now, when they both come in, what happens is they form a very strong bond. So between you, could you link arms, please? So what we have there are 2 amino acids linked together. Okay, Xena, keep on going, please.
Val.
VAL, where are you? In you come, form a bond, please. Link arms, form a bond. Next.
GAG. Glue.
GLU, in you come, form a bond.
Okay.
That's fantastic. Right, can you go down, girls? Can you go down onto the floor so that Xena can see, please? That's fantastic. That's great. Keep going.
UGC. CYS.
Okay, and stop there. Can the rest of you tRNAs just sit down for a second, please? Fantastic. That's really, really, really good. Have a chocolate, guys. Have a chocolate. So what have we actually formed? What have we formed here? Femi, what have we formed? What have we got? What are these, did we say?
Oh, proteins.
Now, these are not proteins, these are
Amino acids.
amino acids. So what do we have here? Very good, I like that. We have a chain of amino acids. What do we get when we put a chain of amino acids together?
Proteins.
Fantastic. Now, the section of DNA we had up here was one small section of a gene. What we formed down here is one small section of a protein called one gene, one protein hypothesis. And it's very hard. Let's go through it again. So from the top, using the correct terminology this time. Francesca, what's the first thing we do? We go to the nucleus.
And copy a strand of mRNA.
Do we copy a strand of mRNA? We copy the strand of?
The DNA.
We copy a strand of DNA into?
Oh, the mRNA.
Into mRNA. And do we use identical bases to the DNA?
No.
We use the?
The
Complementary, excellent. So once we have a complementary basis in the mRNA, where do we take it to? Where do we take it to, Tony? Ribosome. Once it's at the ribosome, Zena, what happens next? These people called?
What is it? Oh, transfer RNA.
What do they do?
They bring in their what was that word?
Amino
acids.
Fantastic. They bring in the amino acids and then they line them up. And what forms between each of the amino acids? What forms between them? Bonds to form a protein. And that is the one gene, one protein hypothesis. For a final time, because it is so hard. In the nucleus, the DNA is copied using the complementary base code. From this, we form messenger RNA, which goes to the ribosome. Transfer RNA molecules bring in what? Amino acids, which then bind to form a protein. Fantastic. This is vital, and your body is doing it at this very moment, millions of times over, all of your body. Does anybody know what we've actually formed there? We formed a chain. Have a look on the board. That's what you are. You form the first part of a very important protein. That protein is called insulin. And it's the thing that is helping you deal with those chocolates that I've just given you. Because without that, you would be in big, big trouble. Thank you very much.